And that takes us now to the next part. This is now the panel discussion of the 2 recorded talks. And the first one was by Hannes Bergmann. So Hannes works at the Friedrich Löffler Institute in Germany. And you remember maybe this morning, if you listened to Crystal Donnelly's talk, she started out with what can you do when you have very sparse data? And this is exactly the question that Hannes, or the problem that Hannes had for African swine fever. And he will tell you about the route they will take. So I thought this was quite interesting. So Hannes, yeah, maybe you give a brief overview before we can then discuss. You're still muted, Hannes? Still muted? Hannes, can you hear me? You are still muted. Your microphone doesn't seem to work. No. So it says you're unmuted, but we don't hear you, or I don't hear you.
I don't hear neither.
Okay.
This is right.
Now we can hear you, I think.
Yes.
The sound from these ones.
Yeah, that's okay.
This works better.
That's good.
Ah, fantastic. All right. That's too much technology in one computer. I didn't cope. All right. You can hear me. You can see me. Thanks very much for the introduction. So I'll just get straight to it. You can see a little screenshot summary of our presentation, and I'll just jump right into the key question, which is, where do you think will African swine fever occur next? And then the most important question is, of course, why do you think this? So to help answer this question, we've developed a participatory risk mapping system that helps us to capture spatially and timely explicit expert opinion about the risk of disease occurrence, particularly for complex transboundary diseases such as African swine fever in this case. So the risk mapping system works by providing participating users with a grid-based interactive map, of which you can see a screenshot here. And you can see also in the screenshot colorful squares and dots, which indicate the location of disease, ASF in this case. And also you can see a fairly simple base map along with the disease information, which already shows you a range of potential risk information that might be considered for disease occurrence, such as you see road networks, you can see distribution patterns of forests or surface water or location of towns, if you want to consider those. But within the interactive map, we provide the participants with a lot of additional information as well. For instance, they can display maps of wild occurrence probability or peak density and other information as well. So upon providing this map, the participants are then asked to predict the occurrence of disease during the next month, and then also to record the underlying risk factors that they considered for each individual selection that they make. And then by the end of the month, we have collected the predictions of the participants, and then we obviously see the truly emerging location of disease, and we can use that to validate the accuracy of the prediction. And so overall, that's how the system works. And with the data that we generate by using the system, we get, for once, obviously, a user-based, readily applicable risk assessment, which we think is very interesting and very useful to complement conventional stochastic or statistical models. And there's a lot of other little quirks in there that more lead towards cognitive science approaches. And so, yeah, we think it's really interesting, and I really hope I could spark your interest, potentially to go back and watch my pre-recorded presentation if you haven't already done so. Or else, I'm really looking forward to hearing any comments or questions you have. Thank you.
Thank you. So, yes, Pauline, you may want to start with a question here.
Thank you, Hannes. I really look at your talk yesterday, indeed, with a high interest, and I was wondering if you could use through simulation, a bit as you did for assessing the added value of the distance to cases as a risk factor to to compare the random allocation of pixels with the one allocated by experts. Could you do a bit like that, but accounting for more factors in order to identify which one were the word is too hard, used without noticing by the experts or by intuition? Because you mentioned that they might use some intuition to decide which pixels are more at risk than others, and maybe it would be feasible to identify which are under these intuitions. I don't know.
Yeah, thanks, Pauline. If I understood you correctly, that's exactly the approach that we've taken with that simple simulation there. Ultimately assigning different probabilities of selecting free cells in the map based on the value of different risk factors. And I've just given you the one example there of distance being obviously a really important one.
To quantify the weight or to prioritize among the different risk factors, the one that seems to best represent what the experts have done.
Yes, yeah. So that's something we want to explore further. We haven't included more risk factors. So you could, for instance, consider another one being distribution of forest, which is something the participants mention quite frequently as a reason why they've chosen a certain cell. But I think also to just basing this approach on probabilities, on single probabilities of selecting cells is a fairly limited approach. But ultimately, it will give us, when we compare it to the random selection as a baseline, it will always give us an indication as to whether a specific factor will increase the probability of selecting the right cells. So it will give us an indication, but I don't think it's a good tool to implement what the participants tell us into a risk model. I think there's other ways of doing that much better, but ultimately it will help us to maybe get a handle on which factors the participants are considering may be useful, if that answers your questions.
Yeah. I see that no one else for the moment raised the hand, so maybe I can ask my second question. You assess the fact that distance to cases could explain or not the way the pixels are chosen, but maybe it could be also used to assess differently the way experts identify the pixels that will be infected, because for the moment, if I have well understood your presentation, it's really a pixel that is detected or that is said that would be at risk. They have to match perfectly, but maybe you would prefer a system that never match perfectly, but is always in the right areas, compared to a system that match perfectly sometimes, but is very wrong in other cases. So did you think about accounting for this in the assessment of the way experts identify risk areas?
Yes, indeed we did. And I totally agree. Using the direct hits that we show in the talk, which is the exact match of the prediction which the occurrence of disease in that same month is obviously a very strict way of measuring accuracy. And like you say, sometimes the prediction will be fairly close to the actual occurrence of disease. And I think, like you suggest, it would be very useful to relax that stringency of the direct hits a little bit, for instance, by placing a kernel onto the predictions as well as the occurrence of disease and measuring the overlap. So you get a bit of a spatial smoothing, and that's something that we have done, and it obviously increases the information that you get out of the system. And then secondly, the timely element, which is in monthly circles at the moment, where participants need to predict for the exact month when the disease will occur. You should probably gain information by, you'd be more, it might be more informative to relax the timing as well. So that means that if a prediction has been made for the next month and the disease doesn't occur in that month, but maybe 2 months later, it would probably still be considered quite a valuable piece of information. Or you could also then, that's another aspect, if you look at the different predictions that were made by different users, and they sort of seem to highlight a certain area where no disease has occurred yet. And there may not be disease occurring in that same month, but it will still highlight that for whatever reasons, the users have considered a certain area to be at high risk, and that would be worth focusing control efforts or surveillance efforts into that area. So relaxing it in a spatial way and in a timely way will probably make the system more applicable if you were to readily just use the user predictions immediately as a risk assessment. Yeah.
Okay, thank you.
Hannes, I was wondering, so you chose experts as your information source, right?
Yeah.
I was wondering, what makes an expert, and is it better to have many experts, you know, a wide range of experts who may not have very specific expertise, or a very narrow range of experts who know a lot about disease spread, or in particular of that disease? So what do you think is the right approach here?
Yeah, Andrea, thanks for that question. That's exactly a key aspect that we're very excited about with this system. So to be honest, I don't actually like the word experts very much. I tend to say knowledgeable person. And with that, I mean, every person you meet on the street has their range of knowledge, their experiences, and even a local farmer in an ASF affected area will have probably quite valuable information about aspects of how the disease behaves on the ground locally. And so along with what you're just saying, and there's some studies that go into the same direction that support this, it's indeed very useful to have a good range of knowledgeable people that have knowledge and experiences in different areas. And then by tapping into that experience collectively, you get a sort of crowd effects potentially by having by combining these pieces of information, and basically by using each of the users is using a certain strategy based on their knowledge and experiences to select or to make the predictions. But the strategies that are being used by the different users are not necessarily the same. And so that's what I was mentioning before with the cognitive science that needs to be applied to the system as well, to really harness the information that we can gather here. So like you said, ultimately we're using knowledgeable people as our model, a black box essentially, but we try to open that black box and see which strategies that they use. You may call them heuristics because they're not spending too much time on making their selections, so they can't make any complicated calculations and estimations. They pick their predicted risk areas based on a few factors they're considering and on their experiences. And that's something that we're also very interested to find out about. And we actually do collect a bit of background information. We provide the participants with questionnaires. So we collect information about their experiences and their knowledge and their education, and the relevant and how this may relate to the information they're providing us in the system. And we haven't analyzed that in detail yet, but that's definitely an important factor as well. Yeah, thanks.
Thanks. No, very fascinating, and obviously a scope for huge future improvements with machine learning and crowdsourcing and all these things. So very interesting.
Thank you.
Yeah, thanks for that. For the sake of time, we need to move on. And our second recorded talk was from Laura Gonzalez Villeta. Laura is at the moment PhD student, I think, at the School of Veterinary Medicine at the University of Surrey in England. Laura, can you tell us first a bit about your study, just a summary for the people who haven't had a chance to listen to your recording? You're still muted as well.
Sorry. Good morning, everyone. Yeah, let me put this. Okay, so the project that I'm working on is aiming to understand and disentangle the complex role that the environment has as a driver of salmonellosis in humans. So what I have done so far is just to estimate the rate of infection, the lambda, with a weather data set for 30 years in the past at a daily resolution. And so this lambda is not fixed. It varies depending on temperature, and it varies from day to day. So we simulated the behavior of Salmonella in the weather with literature, with formulas that are based on lab studies that have fine-tuned the behavior of Salmonella under different temperatures in the main food sources involved in salmonellosis, which is chicken meat and eggs for the moment. This is a very preliminary stage, but so far, as you see in the graph, I have reproduced the infection of salmonellosis in humans by normalizing with the number of residents at a certain size of area. The area size that we used was laboratory postal codes, since it's as a proxy of the location of the patients and keeping the privacy of the patients as well. So the good fitting of the curves confirms that the lambda use is fit for use in the model that I'm building. This model will be a stochastic population-based model using the Poisson distribution, which in a sense will generate the probability of people to get sick due to salmonella transmitted through food at a certain time and location. This model will assume that every person will have a similar diet, similar amount of eggs and chicken eaten, and similar amount of bacteria infected or potentially contaminating these foods. Once the model is polished and validated, it could be used to estimate the burden of salmonellosis in different climate scenarios, such as increased temperature rising due to climate change. So if this has struck your curiosity, please take a look to the whole video, which I provide a few more details, and I'm happy to answer any questions that you may have for me.
Thank you very much. Are there any questions from people from either today or if you had a chance for recording? Let me just check. Elisa, you have a question.
Yeah, thank you for this interesting topic and the flavor that you gave us to listen to your talk. So I was wondering, just the difficulty here is in proposing a relevant set of potential covariates, or is it are there some technical complexities in terms of, I don't know, variable selections, so statistical issues, or which are, in your opinion, the most, yeah, the most, how do you say it, the hottest
challenges.
points? Yeah, the challenges. Yeah.
Yeah. So I think the whole project is a challenge, just in the base that we are trying to know the role of the environment itself. So we are trying to understand something that we kind of is intuitive to us, because we see, as the graph shows, a more amount of cases during the summer peak, like summer months in August and September. So intuitively, we know that there's some influence of weather and especially temperature, but there's no studies that tell us how exactly this influence the incidence. So basically the lack of data to work with, it's hard to model something without knowing the exact behavior. So that's why we're trying to get an approximation on how the environment drives the disease. So in summary, the data is the challenge, the main challenge for me, and also the weather itself it's very complex. We have only model temperature, but it has different variables such as humidity, sunlight, or rainfall. It has so many variables that are influencing one another. So the challenge would be to unify how both variables would affect at the same time. And that will be one of the next approaches to my study. This worked very well with temperature, but what will happen if we sum humidity to the model, how the incidence or the simulation of the incidence
but what about putting the most variables you can, and then so making variable selections, or even using I don't know, yeah, more sophisticated techniques if you have many? You want to add them one by one, or because you can imagine to put them all if you don't know if it's the humidity or I don't know.
So for the moment, like as I said, we have to know how Salmonella behaves in this food stuff and under different weather influences. So I have found this in temperature easily, and I have some kind of hint to how it behaves in humidity, different humidity levels. So that leave us something to work with. So I will start with only these two parameters and see how they combine, how they worked. And if they predict the incidence of the past, only with taking into account temperature and humidity, that means that at least these two are the main drivers, or one of the two most important drivers. And so I'm not intending to include all the weather possibilities, but the most important ones.
Okay. Okay.
Colin, you had a question.
Yes, I'm sorry, I haven't actually watched your full presentation, so I'm not sure how relevant this question is to what you've done, but I'm aware that there's been a bit of a revolution in the detection of human foodborne outbreaks recently because of the use of whole genome sequencing, which has suddenly allowed the fairly easy linking of cases that previously would not have been identified as being associated with each other. I was just wondering, was your work influenced by that? Because my understanding is there have been more outbreaks identified recently because of that capability that didn't exist before.
Yeah, so I'm working basically with big data sets, mainly from years in the past. So the first year that I have records are from 1999 till 2020. So for all these years, I don't have whole genome sequence data. So I'm not including that in my studies. If we will have a big data set with whole genome sequence information, that we could probably easier to detect, to point the outbreaks to the food source. But for the years in the past, I don't have this information. So I'm not working with this.
All right, thanks.
So I have had salmonella infection in the past, so I'm in full support of any study that prevents these things.
Did you know that it was salmonella? Did you get...
Yeah, it was diagnosed, yes.
That's interesting.
One thing, so I know you're focusing on climatic driver for salmonellosis. There's also now a lot of change in diet going on. So, you know, we are constantly told that we should change our diets, and there's this huge vegan or vegetarian drive. And I'm wondering if that would be also something to consider, you know, because I know I saw your model includes, you know, at least the different sources of salmonella infection, so eggs versus pork, whether it would be worth also looking a bit into that, if there's scope in your study to do that.
Yeah, that's an interesting point that I have also considered, mostly because I've noticed that in the UK there are many vegetarian, or there's a very big trend of vegetarian people and vegan. So I'm in contact with one of the main supermarket chain and trying to get consumer data. So this is something definitely in my mind, although the data available is not for so many years in the past, it will be more for recent years. But if they are able to provide the data that I'm at a small resolution scale that I can do some analysis, I will happily include it, yeah.
Okay, brilliant. If there are no other questions, let me conclude the session. Let me thank all the speakers, whether we heard the talks or not, for presenting. It was really interesting. I learned a lot. So yeah, big clap to all the speakers, and I hand over to Elisa to close this session.